Okay, hi again, everyone. Hope you're doing well. So we're on to lecture 8. So we're going to start talking about sensory systems. And today we're going to go through a little bit of the theory. I'm kind of wondering how you're doing. So we kind of rushed through different lectures. I hope all of you are doing all right. There's a lot of material to cover, and I know that. And I'm hoping to use Thursdays to address some of your questions in person. I miss kind of interacting with students. I hope it's the same for you, that you miss interacting with your profs. And I hope that you're doing well. Again, feel free at any point in time using the email that I'm going to show you on the next screen to reach out just to let me know how you're doing. I'm really quite curious. I'm doing all right. We're starting to hear that maybe things are going to be back to normal-ish sometime in May, and we can always hope that there will be a good resolution to all of this. I want you to focus as much as you can during this time on the material that's coming up. Some of it's like really exciting, very interesting. So let's go ahead and dive right into it. So I'm going to start sharing my screen. Just bear with me as I get things set up. All right, so almost there. And let me grab my trusty laser pointer. So again, we're going back to cover some of the major concepts that we need to think a little bit about when we talk about the senses. Some of it is stuff that we've already heard before, and these were lecture notes from Dr. Key. Again, I'm adapting some of the lecture materials from my colleague, Dr. Douglas Tweed, also in physiology. And today we're going to look at the senses, and we'll be using his lecture notes. Again, drop me a line. Let me know, do you like this style of lecture notes better, or would you prefer having the ones that Dr. Key had? And with Dr. Tweed, we always start off with the question of the day. Now, you might be thinking, well, that's obvious. I've heard about 5 senses, and wasn't there a movie with Bruce Willis a while back, The Sixth Sense? So obviously, don't we have 5 senses? So let me try to reframe that in a physiological perspective, and we'll talk about how many senses you have a little bit later on. But interesting question, keep that in the back of your mind. So we're going to go through some of the concepts we already covered in Dr. Key's section with receptors and neurons. And then we'll be talking about transduction, changing a physical stimulus into an electrical signal or something that begins an electrical signal. We'll have to go back and revisit stimuli, how coding in the nervous system actually occurs. Like, how does your brain know that a photon of light represents something? How does it code for that? So we'll go back and discuss that. And then, of course, when we look at that, how do we know about differences between different types of stimuli? And how do we code for those changes in the nervous system using frequency changes or intensity changes? And then we'll get into this signaling change. And then we'll get into some very interesting phenomenon about higher order processing. Your brain likes having patterns. You like having patterns, and so does your brain. And we'll get into that in a moment. So let's start off with looking at these receptors as well as neurons. And remember, every sensory system begins with these receptors. So there are a number of cells, and these don't necessarily have to be neurons, that begin the process of converting whatever signal they're detecting into an electrical signal that we look at as either a postsynaptic change or as an action potential. This conversion of that stimulus, whether it's like touching your skin with receptors on your skin and then the neurons that they're innervating or that they're connected to, or within your eye, all of this is known as transduction. So this receptor that begins this process is going to convert this stimulus energy into receptor potential, and that's found within that receptor cell. And then this receptor may in turn release neurotransmitter onto the next neuron. And we'll see several of examples of that. In fact, especially within the visual system, we'll see examples of this. If this receptor does, though, happen to be a neuron, it could actually begin the process of firing action potentials directly. And typically we see that within the cutaneous system, the neurons that innervate your skin, we often see that as well. And we'll get to that, so you'll just have to be a little bit patient. So each of these receptors has what is known as an adequate stimulus, and this is the form of energy that is going to activate or stimulate that receptor the best, and it's the most responsive to that particular stimulus. It could respond to other forms of stimuli. So for example, in your skin, another really good example, thermoreceptors, as the name suggests, the receptors in your skin that use thermoreceptors, they're the most responsive to temperature, but they can give some information about pressure as well, but they're the most sensitive. Their adequate stimulus is actually temperature. Hopefully that makes some sense to you. And we need to have that vocabulary before we can begin. So receptors are also going to be classified according to their adequate stimulus, what actually activates them the best. So for example, chemoreceptors respond best to specific molecules or ions, and they could be chemoreceptors for glucose, or they could be chemoreceptors for oxygen, changes in acidity or pH, hydrogen ions. These could stimulate and be the adequate stimulus for chemoreceptors. Mechanoreceptors, again, usually respond to changes in mechanical energy, whether it's like pressure on the skin or vibration. They could also respond to gravity by having different structures pushed down on them, as well as sound. And so they're responding to some kind of mechanical force being applied to their receptors. We've already talked about thermoreceptors. They're going to respond best, and their adequate stimulus will be changes in temperature. And photoreceptors, of course, again, responding to light, and that would be their adequate stimulus. Some of these receptors are incredibly sensitive, and these receptors all have a threshold. And this is the weakest stimulus, the lowest stimulus that it can detect. Now, some of our receptors, as the title of this slide suggests, are incredibly sensitive. So some photoreceptors, which we will identify later, we have two different sets, rods and cones. Some of them are able to detect like one single photon of light. And so they're very sensitive. Chemoreceptors, again, are also very sensitive, and they can detect like even one odorant molecule. That doesn't necessarily mean that you'll be able to actually smell something or see something if you get one photoreceptor being activated, because the perceptual threshold is different. The perceptual threshold is the weakest stimulant, so the lowest level of light, not just one photon, but the lowest level of light that you can actually see as of being dark, going from dark into light, for example. All right, so there's a difference between a threshold of a receptor versus a perceptual threshold. So what activates the receptor, that's the receptor threshold, and what you can detect, consciously detect, perceptually detect is known as a perceptual threshold. Hoping that is clear for you. Again, at any point in time, just as a reminder, it's always okay to just stop and pause. And if you do need the textbook at the very end of this PDF file, you'll also be able to see some of the suggested readings that you might want to just go back and look at. You don't need to, but that option is there for you. So here again is sort of how generally these sensory systems are arranged. So we usually have some sort of primary system, secondary groups of neurons, and tertiary neurons. And the first neurons within this system, and these could be either neurons or they could be different receptor cells. These are called the primary sensory neurons, okay? So these are the first. So either the receptors or the neurons that come directly after. And we've talked about that case earlier. So the primary sensory neurons, the ones here in purple on the far left, will then make synapses onto these secondary neurons that you see here in the middle. And you can see that many primary neurons are making synapses with fewer secondary neurons, in the same case here, same case down here. And these are going to, in turn, synapse onto a tertiary or a third order neuron. Okay, so at each of these stages, the presynaptic cells can contact any one of the postsynaptic cells. And what we're seeing here from primary to secondary, and even from secondary to tertiary, we are getting what is known as convergence. Many different neurons are converging onto smaller groups of neurons along the way. So they are collecting data, in other words, from groups of neurons, and that's that phenomenon of convergence. Again, something to maybe circle highlight, and then think a little bit about for when you're going back to review and to study. So sensory neurons can carry information about many different aspects of the stimulus. So just because something is touching your skin, like you can actually get lots of information from that. And this gets into some different terminology that we've actually learned about before from Dr. Key's section as well. And just to go back and review it here with Dr. Tweed's notes. You're gonna have to excuse me, I'm kind of a little bit, it's a little dry in my house today, so I'm kind of having to cough a little bit. So apologies on that. So one aspect of all of this is about different aspects. So one is the stimulus modality. So whether it's a light, whether it's a sound, or whether it's a touch, different sensory systems are geared best to be able to respond to that sensory modality, okay? And again, some of these terms can be a little bit similar, similar sounding for you as well. So we go back to this idea of these labeled lines. And we talked about these labeled lines, that generally speaking, things in the visual pathway that are normally there to respond to photons of light, that activity is going to be signaling into the visual pathway, but we can actually fool that system, and that's because of these labeled line pathways. So the modality, the stimuli are revealed by which axons are actually carrying the signal, and these are labeled lines. These are like direct pipes, in other words, or subway lines. So like everything on the Danforth is only going to go along the Danforth or line number 2. And so activity in axons that are found within the visual pathway usually means that there's been some kind of light activity or some kind of photons that are activating this labeled line, a specific line. And activity in neurons in the auditory pathway means that you're usually detecting sound. Now, can you fool the system? Yes, and we talked about like getting a really hard shock to the eye. Normally it's there to detect light, but actually activating that pathway or those neurons can actually cause a phenomenon where you get like a light aura. And again, that's because it's normally interpreted by the brain as carrying information on this labeled line by photons or visual signals. Now, and I'm also going to get into this phenomenon as well. So earlier, we had talked about population coding, and we had also talked about frequency coding, and this was from the slides that were given to you by Dr. Key, and he normally talks about this during his lectures. And then we are currently on the set of notes by Dr. Tweed. And here, Dr. Tweed's, I kind of prefer the way in which Dr. Tweed kind of interprets this a little bit more. Respect both of them, respect. So here's where I'm going to actually say that I give a little bit of nod. Dr. Tweed's interpretation a little bit better for me. How do groups of neurons represent intensity? And they can do it in 2 different ways. So if I'm pushing down lightly on the skin versus like really hard on the skin, that stronger stimulus may activate more neurons, and it makes sense. I'm pushing down harder, it's depressing more of the skin area or pushing down on more of the skin. And the way that the skin will represent this signal will be that more of these neurons will become activated, and more neurons being activated says that the brain recognizes an increase in more neurons responding as population coding. And it's not just the different ratios, but the sheer number of neurons responding allows for population coding. Hope that makes it a little bit more clear. I know some of you had some questions about that. In addition, the neurons that are being stimulated by a stronger sort of push down on the skin may actually make the individual neurons that are responding fire at a higher or faster rate. And this is also known as frequency coding. So you can represent intensity by the number of neurons that are going to be activated, as well as pressing down harder will make those same neurons actually fire more. So both the number as well as the frequency of individual neurons being activated will change. And that's how your brain interprets changes in intensity. So harder pushes, both of these phenomena will be occurring. Again, it's always a good idea to stop, pause, and make sure that you're comfortable. Good slide for you to know. And these individuals, wonderful individuals in the neurophysiology platform within neuroscience. So, I hope that at some point you get to chat a little bit with them. So how do we signal, now that we know that these receptors can signal for changes in intensity, and they have all of this convergence, how do they actually signal for change? Because we need to know and have both properties. So we need both convergence, the fact that we get many neurons synapsing onto fewer secondary, and those secondary synapse onto fewer and fewer tertiary. That convergence and the changes in intensity are things that we can actually use now as ideas to signal change. So here's the first one. And again, we're going to go and take a look. So here's a stimulus. So this is baseline. Then we suddenly apply a stimulus, and it could be like suddenly increasing pressure and then keeping it on. And this would be where it's keeping it on. How would the neuron underneath that, or the receptor underneath that patch of skin, respond to this? And you'll see that normally when there's no activation, no stimulus, it's at rest, there is no response, and each of these lines, these vertical hashes, represent an action potential and spiking activity. And you'll see that as soon as we stimulate, we get increased activity. In fact, we get a burst of action potentials. And now you know the cellular mechanism behind that burst of action potentials. It's not just the voltage-gated sodium channels, but also the voltage-gated potassium channels that allow them to come back down. So that spiking activity becomes important here. And then you can see that in the continued presence of this stimulus, what do you see happening to this neuron that is responding to that stimulus? Does it keep firing? So the stimulus is still there, but does it keep firing over here? Can't hear you. You can shout at the screen a little louder. Actually, you'd have to be shouting through time as well, because I'm not sure when you're actually going to watch this, but I'm hoping that you can all see that there is no more activation of that neuron. So it's not just about the stimulus, but it's also about how the stimulus is changing through time. It's just a constant stimulus. It's staying the same. It's there all the time. And we can also see that it activates the receptor. But if it stays on there and it stays at the same level, we're not really going to see a change. So when a stimulus suddenly increases or decreases, and here we're seeing an increase, these receptors and the neurons that might be synapsing with them respond briefly and then fall silent again. And so these cells, these receptors, are signaling for changes in the stimulus, not these steady levels. And you can see that very clearly. This is a steady level and there's no response. It signaled only when there was a change or a stimulus being applied. And different cell types, and they can vary in your skin, will have different dynamic properties. So the one that we looked at in the previous slide was this example, and you can go back if you want. If you don't trust me, you can go back, it's okay. And when it's responding to this stimulus here, it exhibits what we call a phasic response. And we can also say that it's rapidly adapting. So either term, and they're used interchangeably, phasic or rapidly adapting receptor cells respond briefly to any of these changes in the input or the stimulus, and then they stop firing, which is what you are seeing here. So they're really good at signaling changes to an environment. So change on your skin, for example. We also have different types of receptor cells in your skin, in your eye, in different places. So any of the sensory systems. We also have what are known as tonic responding cells. And these are also slowly adapting cells. And these cells maintain their activity even when the stimulus is not changing. So let me take you through a couple of things in this example down here at the bottom. So here's an example of a cell that is responding to a stimulus. Here is baseline. And you can see that even before any activation of that receptive cell, that we still have a little bit of baseline firing. So just at rest, without any stimulus, it's already firing action potentials. So in this stimulus now, we see that we get a burst of activity. So it knows that something has happened and it's higher than at baseline. Because again, if it was the same as baseline, how would your body know that there was something occurring, that a stimulus was activating it? and we can see a burst of activity, but as long as that stimulus is there, unlike the phasic situation where it stopped firing, and it's only there to signal that a change is occurring, it actually will continue to fire as well. And then they tell you, or they tell the brain, about a continued persistent level of the stimulus still being there. So we have these two different cell types that are responding in very different ways. Now, that was a general case. And what we're going to be looking at starting in the next lecture series is the visual system. So we need to start adapting a little bit to the visual system. And many of these retinal cells, the ones that are involved in the visual pathway, are actually responding phasically. And then we're going to do a couple of exercises to prove to ourselves that this indeed is the response. So your eye is really attuned to changes in the visual world. So you could be staring at a wall and it's not doing very much, but if you suddenly see a spider moving across it, it's responding to like movement or a change in the visual world that it is observing. So you wave your hand to catch someone's attention. So if I'm sitting up here in the corner and I'm not really doing very much, and you're not really paying too much attention to what's happening, and I'm talking a little bit slower and your attention is kind of waning a little bit, and we've all had this experience, and I'm staying pretty still. If I wanted to suddenly grab your attention, I might start waving because this will catch your attention because it's activating the phasic responding cells within the retina of your friend. Now in the next slide, we're actually going to see what happens to your retina as it adapts. So if you take a close look, you're going to look at the X in the middle. There are little pink clouds all around. And then if you focus only on the X and you don't let your eye shift around, and you have to keep your concentration there, but if you kind of look, you'll start to see these little pinkish clouds all around the edges start to disappear. So look at the X right in the middle and just keep staring at that X. And don't let your eyes move around too much. You will slowly start to see these pink clouds on the periphery disappear. And you can do that exercise at any point. Staring at a screen, it's not a bad thing. And these phasic signals actually are useful for us because most of the time our world is fairly stable. And this is, again, useful. Like, we don't like having surprises. So, you know, those scary movies where everything looks like it's nice and calm and then suddenly something jumps out at you. And that's because, again, of phasic signals, and interruption and the routine types of patterns. All of those are kind of interrelated. So it's better for your brain because, again, it wants to be as energy efficient, and we learned that in the last lecture, to report on changes rather than just saying everything is the same as it was before. Everything was the same as it was before. So if you're gathering information about changes, you don't want to continually hear, yep, it's sunny. 5 seconds later, it's still sunny, still sunny. It's better if you get a change like it's no longer sunny, it's actually now cloudy. So these types of changes through time, over the course of like one moment to the next, these are known as temporal changes. And again, just a little bit of humor for you. Does a watch pot actually boil? Of course it does. But it just seems like we're waiting for that time to end. So those are temporal changes. And those phasic receptors in our eyes or in our sensory systems are really good at phasic changes. But we can also use these receptors in our eyes and on our skin and in other places in the sensory system to tell us about spatial changes. Okay, so not just over time, but also like in terms of edge detection and other things. So spatial changes are a little bit different. So they're the differences that occur between neighboring regions in space. So being able to detect where your laptop is stopping, or where the edge of the screen is stopping. Let's see if I can actually point to it. So here is the edge of the screen that you see over here. And then below it, you'll actually see the rest of the sheet that you're looking at. And so those differences, those hard edges, are ones that we want to be able to detect. And these spatial changes are also known as contrast. And where we have the strongest contrast, so for example, darker versus lighter, the difference between those two different types of spatial changes is what we call an edge. And this is where the strongest contrast is. So again, going back to that weather station analogy, so neighboring stations over here, for example, it's still dark, it's still dark, it's still dark, it's still dark. But here, if we went from this station to the next, we would go from dark to suddenly being lighter. And this edge actually gives us a way to notice a change spatially in our environment. So the previous example, basic changes or phasic responses allow for temporal changes. And now we're looking at spatial changes based on these edge and edge detection. And how does that actually occur? How does your sensory nervous system, whether in your skin or in your eye, actually allow for edge detection? How does it know where something is like really light, the border between like dark and light? You can see that pretty clearly here. You can see what is light and what is dark. How do you know light to dark? And we use different parts of the sensory system. So again, it's been about 20 minutes since we started, but if you go back, this is sort of that 3-layer system. And we have primary, and we have secondary, and then we have tertiary. So one first order, second order, third order. It was given to you sort of more horizontally. And here's the thing, when we talk about having convergence, you don't really see it overtly here. It does exist, it's just drawn for you so that it's easier to understand. So if you're asking, does convergence also occur? It does, but let's just keep it simple for the time being. So we have 3 different neurons in this circuit: primary, secondary, tertiary, primary, secondary, tertiary. That's another sort of strip. And then between these second order neurons onto the tertiary, they will also send not just one axon, but they'll send a branch of an axon laterally to the sides to inhibit their neighboring tertiary neuron. And this is going to be known as lateral inhibition. And this is the way that we get edge detection. The next few slides are going to take us through this process. And that means that if we excite this pathway, it's going to activate this next neuron down, but it's also going to send a projection. As this gets activated, it activates this one, but this branch will actually inhibit their neighbors. So this lateral inhibition means that that one secondary will be inhibiting their neighbors or inhibit the cells that the neighbors would normally be exciting. So again, this means that we would have these inhibitory connections as well. Okay, so these other pathways here in green, they're all excitatory. So activation here activates the secondary, secondary activates the tertiary, but this branch will actually inhibit their lateral neighbor. Again, a great place for you to stop if things are kind of getting to a point where you need to stop, think, go back and revisit your notes. And if you're okay, we'll just go on to the next slide. So here is a scenario that I'm going to take you through step by step. And I really love the notes that Dr. Tweed has put out for all of you. And this is a great example of lateral inhibition or edge detection using lateral inhibition. So let's take a patch of skin. So here's this patch of skin, and this is the normal skin here, the top layer. Here is the primary, secondary. We don't see the tertiary, but it is there. And we have an object that's actually pressing down, depressing on the skin, and it's actually shown as a little divot here. And there's a hard edge between where there's contact, where my finger is pushing down on my skin, versus the area of skin right beside it that isn't being pressed down. So how do I know like how big this is and how much of an edge it's actually creating? So if an object is pressing down on the skin, these primary and secondary neurons in the area of contact, so here's that area of contact, are activated, and these over here are not activated. Again, pretty simple, right? Do you even need lateral inhibition? Well, yes and no, and we're going to get to that in a moment. Again, a really good series of slides for you to know. So these are not going to be activated. They're still at baseline. And the ones here in the middle are more activated. Again, I know you're all with me so far, so if you can indulge me, I'm going to go on to the next slide. Here's where we're going to start integrating a couple of really basic concepts that we've learned about in the previous slides. And again, I've gone a little bit slower today to make sure that you understand everything. So we're going to consider the ones here, because here is that edge. So here is where a station would be signaling there's something going on, and everything over here would be nothing's going on. So again, now we're looking at the tertiary neurons as well. Here are those side branches that we talked about. In red, it means that they're inhibiting. Any green pathways means that they are exciting. And so here is this object. So let's examine this one here. So cell A, so if we had the object pushing down, and it's right at the edge, it's right at the boundary, that object that's pushing down on the skin would activate this primary neuron. And in turn, it would activate the secondary neuron. The secondary neuron would then also powerfully excite cell E. That part hopefully makes sense. It's directly being stimulated. And you're all with me so far, right? Can't hear you, but I'll assume that you are. So if we also look at the lateral inhibition, and it actually sends out 2 projections to its nearby neighbors. It will send out projections to D as well as to F, and these are all tertiary neurons down here at the bottom. So while this has been excited, this is now sending out signals to its nearby neighbors. These are all just sort of at baseline, and so this is going to inhibit the nearby neighbors D as well as F. So cell B, the one over here, is actually just at rest, so it's already less active anyways than A. So its effects are not going to be as pronounced on this particular cell. So it's not going to be able to inhibit it as much, right? So it's not being stimulated, so it can't laterally inhibit cell E as much as it could. And so its effects are overall weaker. And so it is going to excite F, and it's not going to really do much in terms of activating E or inhibiting E or G. So that means out of all of these cells in this area, E is the one that is going to be much more active than D, because it is still laterally inhibiting this one. It's not receiving too much inhibition here, but again, these other ones, they're equally canceling each other out because they're getting these lateral things. This one is less active, so it's not as able. So this is, out of all of the ones here, the one that is going to be the most strongly excited. Hopefully that makes sense. And it's good to kind of go back and use sort of the diagram and re-listen to this part again to make sure that you understand about this lateral inhibition, why this one is less active, and that means that this pathway is also less active, which is why it's in a lighter color, and because these are all being stimulated to equal extents, and because this is the only one where the inhibition is weaker coming from cell B, this is going to be the strongest sort of signal. So that means that in comparison to all of its neighbors, it's going to know that it's right at the edge, okay? Or your body will be able to interpret it as being right at the edge. And again, I've just mentioned this as well for cells C, D, G, and H, because they're not on the edges, they're going to actually be canceling each other out. So they're still getting the strong signal or the weak signal. They're still sending out their lateral inhibition. So these tertiary cells, and this again is really the take-home message, these tertiary cells are the ones that integrate all that information, because remember we talked about that earlier. They're integrating all of this information, and they're the ones that are ultimately going to signal that there is a difference in terms of an edge, or that there's a contrast in the sensory input that is coming in. And these are only at these places between E and F where there's a clear difference in the signaling out of all of these different populations down here at the bottom. And I'm hoping that if you spend the time a little bit and go over this series of slides, that you'll understand why this pathway is the way that it is. And again, for the following Thursday, it might be something that you want to go over in some more detail. All right, so if we're okay, let's talk a little bit about higher order processing and how your brain actually interprets all of this information and how it actually uses this information about edges and how it actually tries to process it under normal circumstances and why we can actually fool the brain. So most of these sensory pathways, as I mentioned to you earlier, run through this structure in the orange that you see here, the thalamus. And do you remember what structure that was called? Without looking back in your notes. And if you remember that this was found in the diencephalon, so we also had the hypothalamus, we had the thalamus, we had the pituitary, there's another organ system there as well, the pineal gland, so hopefully that's a review for you. So in the diencephalon, an area known as the thalamus acts as the central relay pathway. So this is the pathway where it's almost near the center, the middle of the brain overall, and it will send information out to the different parts of the cortex that are involved in somatosensory information processing in this teal strip. There is auditory processing, the part of the cortex that's involved in trying to make sense of all the sound. The back of the head, the visual processing area. From here, all of the sensory information will go out to all of these other areas, including ones that aren't shown here. And the olfactory pathway is the only one that bypasses the thalamus. Their neurons don't make synapses here. They actually will go directly to the olfactory pathway. And then the equilibrium pathway will project mainly to the cerebellum. So our senses have evolved to guide our behavior in a lot of ways and to steer us away from danger as well as from situations that are going to be harmful to us. They want us to have something to eat, toward food and shelter, toward making sure that we're comfortable and having a mate. And the big part of this is deducing what's happening and to be able to identify different patterns. That's actually fairly difficult, but your brain does an amazing job because sometimes that information isn't really complete. And in fact, we know that that's a hard job because that's what we do in a lot of these multiple choice test questions. We ask you to kind of find the next part of that multiple choice. And we know that's a hard thing that we're asking you to do. And sometimes it is a little bit ambiguous. And sometimes your brain does have to infer that information. So it takes the most out of that information possible, but it's able to like quickly process. I don't have to write what's up here. You've identified this many different times, and probably many of you are identifying this sort of as unbaked bread. Again, really popular during this pandemic, or you're able to identify this without any issue as being popsicles of different colors, and you can even imagine these different flavors all through inference. Like I didn't have to tell you, you're able to infer and process this. And because of that, our brain is actually easily fooled. If you look at this, this is a sort of a flat quadrilateral. Here's another flat quadrilateral, and here is a third flat quadrilateral. But if you join all these flat quadrilaterals together, you think that this object, the way that it is shaded, is actually a three-dimensional object. But of course, it is really still flat, and it's just all joined together. And it's because these shapes and the way that they're organized, and because you've seen these shapes before in the real world, your brain is interpreting this flat 2D signal as actually being three-dimensional and floating off the page a little bit. And that's because your brain knows that these quadrilaterals should be organized this way. And as a result, it's inferring that this shape is three-dimensional, even though obviously it's not sticking out at us. In the same way, if we look at the Kaneza illusion, you can actually see that there look like there are two overlapping triangles. So this inverted triangle and then this other triangle, it's filling in some of those different gaps. There really are no triangles on here. There's sort of a V here, a V here, a V here, and then V's in these edges. And your brain is just looking at this as a series of coincidences, and it's finding a pattern, it's finding an identifiable pattern, and it's trying to fill in some of those different gaps. And it's assuming that the only way that this can occur is if there's a white triangle right at the beginning. So I've also decided to maybe start posting some different practice questions for you, just so that you get a little bit more of a heads up in terms of what to expect for both the mini test, and I'm still a little bit, I'm still getting some guidance from Dr. Wong about that, so I'll get back to you once I have a little bit more information. as well as for the midterm, this is the way that I'm likely going to be asking you these different questions. So I'll leave this up here. The PDF file is also there for you. It's a good idea from today's lecture to go back over phasic and tonic responses and the different mechanisms that are used by the skin to respond to changes in intensity. And do you know the difference, in other words, between population versus frequency coding? So for Dr. Tweed's section, he's always kind of really nice about laying out the readings in these different versions of Silverthorne. And if you're interested in going and reading a little bit more, these are the pages that are recommended for you. So I think that's it. I think I'm going to stop sharing right now. Apologies on sort of coughing a little bit more than it normally would, but it's also just been a lot of lecturing over the last little while. So, I'm hoping that will disappear soon. I'm hoping that you're also taking it slowly and day by day, and that you are still enjoying the process of learning about different things. It's hard to do that if you're just sitting there and just doing all of these at one time. So find times where you can just do 2 or 3 lectures at a time and enjoy physiology if you can. Don't look at it as just studying. Take good care of your physical, mental, and emotional well-being. Stay healthy, keep those synapses active, and I'll see you in the next lecture, somewhere down there. All right, take care, everyone.